I'll start with introduction of the jury members. Probably I'll start from those of us who are online in alphabetic order. Dr. Alexander Bagayev is currently Vice President of Product Development at Boston Gene, a company that works in the area of personalized oncology. Dr. Bagayev got his degree from the biological faculty of Moscow State University, and then defended his candidate, PhD, basically, thesis at the Institute of Immunology here in Moscow in 2021. His research, his experimental research focused on the role of various types of cells in anti-tumor immune response. And after that, he joined Boston Gene, recently established Boston Gene here in Moscow, first as a specialist in immunology, and then he led the immunology team and finally got the position of the vice president. And he has co-authored a fair number of publications, the latest one I've just seen, Dr. Bagayev. Dr. Grigory Yefimov is currently Senior Scientific Director of Immunity Cell Therapy at Miltenyi Biotech. He graduated from the People's Friendship University in Moscow and got his PhD degree at the Engelhardt Institute of Molecular Biology. And then he led the Transplantation Immunology Group in the National Research Center for Hematology, and later the Department of Biomedical Technology there. And I don't know him personally, but a fair number of my former students actually worked in this group. And currently, Dr. Yefimov focuses on targets for T-cell therapy in liquid cancers and optimization of T-cell therapy specifically for optimization of T-cell therapy for leukemia relapse and cytomegalovirus infection. And he has made quite an extensive contribution to the area of his publications. Professor Richard Neher is, Dr. Richard Neher is Associate Professor at the University of Basel. He graduated from the Ludwig Maximilians University in Munich, and then got his PhD degree there. Then he has spent several years at the Kavli Institute for Theoretical Physics, and then got a position at the Max Planck Institute for Biology in Tübingen. And he has started his career as a physicist, but then shifted his research towards evolution, specifically evolution of viruses and bacteria. And well, we all know that epidemiology and evolution of viruses was extremely popular during the latest 3 years, and he has made important contribution to the molecular evolution and epidemiology of pathogens such as influenza virus, human immunodeficiency virus, and of course, the coronavirus. He had got several awards. He had NRC Starters Grant some years ago, and Arches Award of the General Secretary of Science Education, and the Open Science Prize. Now, the jury members here are Dr. Dmitry Ivankov, Assistant Professor at Caltech. He has got his degree in physics in Moscow Institute of Science and Technology, and then defended Candidate of Science degree in biophysics specifically in the area of protein folding. And I think your first advisor was Professor Finkelstein, was it? Yeah, in the group of Professor Finkelstein. And then he worked in various areas of computational biology at Georgia Tech, Technical University of Munich, and Center for Genomic Regulation in Barcelona, and the Institute of Science and Technology, Austrian Institute of Science and Technology. And he continues working on protein folding, but now with an evolutionary slant because he studies epistatic effects on mutations on protein structure. And he has, again, I mean, like all our jury members, he has published a lot of important papers in his research area. Dr. Evgeniia Khrameeva, also an assistant professor at Caltech. She graduated from the Department of Bioengineering and Bioinformatics at the Moscow State University, and then defended her candidate of science degree at the Institute for Information Transmission Problems, and I think I was a supervisor, or was it Professor Mironov? It was myself. Both. Yeah, Professor Mironov and myself were her advisors, and then she worked for like various periods of time in Gießen University, in Munich, in Technical University of Munich, in Shanghai, in the group of Professor Khatovich, the Partner Institute, Shanghai, and Max Planck, and also in Leiden. In Leiden, probably it was when she was a student. And then she joined Caltech first as a researcher, and now, as I've said, she's an assistant professor here. And her research interests actually were shifting because she has started working on alternative splicing and then moved towards transcriptomics and then towards metabolomics and lipidomics and then towards chromatin architecture. And now she has research projects in almost all of these areas. She's a PI of several research grants. And well, just recently she got, like a week ago, she got a Caltech award for as the best teacher. Myself, chair of the jury, Mikhail Gelfand. Well, I'll skip my biography. It's not that interesting. My research areas are mainly in various. My research interests are in various areas of molecular evolution, specifically in comparative genomics of bacteria and systems biology, but again, systems biology of eukaryotes, but again, looked at through the lens of molecular evolution. I teach a number of courses here. Basically, that's it. I'm fellow of ISCB, just for the record. Dr. Georgy Bazykin, he has graduated from Moscow State University and then got his PhD degree in Princeton University in ecology and evolution. And then he won a grant, Startas grant, and returned from the Russian Academy of Science and established the sector of molecular evolution at the Institute for Information Transmission Problems. And since 2016, he is also a professor at Caltech, and his research interests are mainly, again, in molecular evolution, specifically in just sorting out various modes of selection. He has developed a number of new methods for identification of positive selection. And again, recently, he has become quite famous here in Russia because his group was actually the first and probably the only one studying epidemiology of SARS-CoV-2, specifically in Russia, and he has made an important contribution not only to science but also to policy making. And currently, Professor Bazykin is visiting science in Harvard Medical School, following some unfortunate events beyond his control. Okay, that's it. So here are our jury, and we can start with talk. So Evgeniia, please, you have about 40 minutes.
Okay. Hello, everybody. Thank you so much for being here today. So today I'm going to tell about my PhD thesis, which is named Evolutionary Analysis of Intrahost Interaction between Pathogens and Adaptive Immunity. So the arm race between host and pathogen has a really long history and almost started from the origin of life. And it's often described by the famous phrase which Red Queen said to Alice in a Lewis Carroll book, which it takes all the running you can do to keep in the same place. So this interaction between host and pathogens can be considered from a macroevolutionary scale, where pathogens developed a huge diversity of different mechanisms how to effectively invade cell hosts and imperceptibly reproduce it. And from the other side of this universe, her host developed complex and beautiful systems of innate and adaptive immunity. However, this arm race is going on a really shorter time scales, on a microevolutionary time scales inside the single host. So usually pathogens have a really short generation time, and this allows them to accumulate some changes which escape them from immune recognition. And from the other side, adaptive immunity possesses some nice mechanisms to for faster action for such changes. So in my thesis, I'm focusing on this microevolutionary scale of this host pathogen interaction inside a single host. So before the start, let's look on some mechanisms how this interaction between pathogens and adaptive immunity can go. So the whole idea of adaptive immunity is in a huge pre-generated diversity of T and B cell receptors. And this diversity is actually so excessive that most of these receptors will never bind their specific antigens. However, for any pathogen, there would be some set of pathogenic molecules which can be recognized by these specific receptors and make the adaptive immune response to work. The recognition actually happens differently when B cells recognize intact soluble antigens, which can be the pathogens by itself, like viral particles or bacteria cells, or or it can be a live product of pathogens. The T cell recognition is trickier. It relies on presented processed antigens on MHC molecules, which are placed on the surface of antigen-presenting cells. Any one way or another, after this specific recognition, the corresponding T or B cell become activated and expands, producing a clone. So this the amount of this specific receptor increases in several magnitudes and successfully eliminate the pathogen usually. The co-evolution starts here when the pathogen changes its antigen in such a way that specific immune receptor cannot recognize it anymore. So here there is two mechanisms how adaptive immune response can react on such changes. It obviously can involve new T or B cell clones, which will recognize some other parts of pathogen or will recognize this changed antigen. However, such mechanism and new involvement requires time because it's not easy task to find specific receptor from this huge diversity. So in the case of B cell immune response, there is another beautiful mechanism called affinity maturation, when initial specific B cell receptor can adapt and evolve right during the time of the infection and can restore the specificity and binding to the antigen. So, of course, this is a really shortened scheme of how pathogen and adaptive immunity can interact inside the single host. And my work considers two parts of this scheme. Actually, the first chapter is devoted to the study of evolution of B-cell immune repertoires and adaptation of B-cell immune response. And the second chapter considers the system from the side of the pathogen on the example of a SARS-CoV-2 virus. So let's proceed and go to the first chapter, which is devoted to the evolution of B-cell repertoires in healthy individuals. So, and first, let's remind us a little bit about the process of affinity maturation and how it proceeds. So after the initial recognition of the B cell of its antigen, it becomes activated, it goes to the germinal centers of the lymph nodes and become involved in cycles of somatic hypermutations. So these cycles have two phases. The phases of mutations and selection. So mutations occurs in a dark zone of germinal centers where the specific enzyme called AID introduces mutations in the gene of B cell receptor. And such mutations, which are called somatic hypermutations, they can differently affect the binding affinity of B cell receptor to its antigen. So to check this effect, the B cell goes to the light zone, and this is how the selection occurs. The B cell tries to catch the antigen, which is present here, and bind it to its BCR, internalize it, and present it on MHC class II molecules to T helper cells, which are also present there, and to get the survival signal from T helper cells. So if the mutation were neutral or beneficial, the B cell can solve this task and catch the antigen and get the survival signal, and it can go to the next cycles of proliferation and somatic hypermutations. However, if the mutation appeared to be deleterious, such B cell cannot get the antigen, and it can get the survival signal, and it goes to the apoptosis. So as a result, the lineage of B cell, it's not a similar clone of B cell receptors, it's a diversified lineage of B cell receptors. And somatic hypermutations accumulates with time, and actually the average affinity to the antigen in the lineage increases with time as well. So for the best of my knowledge, there are not so many studies considering the B cell immune repertoires from the evolutionary point of view and phylogenetically point of view. And those studies, which I know, they are mostly done on repertoires under some really active stimulation. It's repertoires after the vaccination or repertoires of chronically HIV infected individuals. So authors of these studies developed nice approaches to detect selection in these phylogenetic lineages, and they revealed a strong signature of positive selection in vaccine-responding lineages and in lineages evolving together with the HIV virus during the chronic infection. However, it's not so many, there is no so many information about how B cell immune repertoires evolve in usual conditions in healthy individuals. So this is why in our study we considered B cell repertoires of healthy volunteers. We have 4 volunteers actually, and immune repertoires were obtained from the peripheral blood of these volunteers, and it was sampled 3 times within a year with different distance between time points. And then peripheral blood B cells from it were sorted on 3 subsets: memory B cells, plasmablasts, and plasma cells. And after immune repertoires were obtained, my part was focused on the most abundant clonal lineages, which were revealed by joining of immune repertoires from 3 time points and 3 cellular subsets. And I focused on lineages which contained at least 20 unique clone types to make a phylogenetic analysis possible. So first of all, I wondered how these lineages are composed. So I considered the fractions of different cell subsets and isotypes of B-cell receptor in the lineage, and revealed that in all 4 volunteers there were 2 different distinct clusters of clonal lineages. And we called them HBMM cluster from high B memory cells and LBMM cluster from low B memory cells. So already, as you can understand, they are dramatically different in their composition. So when HBMM cluster were mostly composed from memory B cells with the unswitched IgM isotype, the LBMM cluster was composed from antibody secreting subsets and had mostly switched IgG and IgA as a type, indicating that they involved in some ongoing immune response. So these 2 clusters also dramatically differed in their dynamics. When HBMM cluster had more or less constant repertoire frequency for time, in LBMM cluster we observed some huge increase of repertoire frequency in some time point. And this increase was different between different lineages and between different patients. So it was not simultaneous. So to compare this computationally, we constructed a metric called persistence, which is close to 1 when the dynamics of the lineage is more or less constant for time, and is close to 0 when there is some burst of repertoire frequency in some time point. So it appears that the composition of the persistence of the lineage is strongly associated with this composition. So the more memory B cells the lineage has, the more persistent dynamic it also has. So next, we performed phylogenetic analysis on these lineages and developed such a scheme. So B-cell clone lineage starts its evolution from the germline sequence, so from germline VDG genes, from which its B-cell receptor was originally constructed. However, if we consider for a first node of the lineage phylogeny, which we sample, it can be distant from the germline sequence for some numbers of somatic hypermutations. And it actually depends on how we sampled these observed clonotypes from the real lineage. So if we sample clonotypes regardless from the position of the clonotype on the real phylogeny, this MRCA sequence can almost match the germline or can be very close to it. However, if we for some reason sample clonotypes just for a single clade of a tree, this GMRCA distance can be much larger. So actually it's right the case what we observed in LBMM lineages, when in HBMM lineages its MRCA sequence almost matched the germline. In LBMM lineages it was much larger, and in addition to it, LBMM lineages, their clonotypes were much more genetically close to each other, which was reflected in a mean pairwise distance between clonotypes. So this difference between 2 clusters can be observed even by eye. So here are 2 examples. This is an example of HBMM lineage, and this is a common example of LBMM lineage. So we see here that lineage is not persistent, came from a single type point, and clonotypes here sits on much shorter phylogenetic branches than here, and the most recent common ancestor of the lineage is distant from the germline sequence, so some part of evolution of this real lineage phylogeny is hidden from our eyes because we sample just this part of its tree, and the opposite picture in HBMM lineages. So together with all these observations, we also revealed that in the most albumin lineages, there was at least one B-memory clonotype which were detected in the time point prior to the main lineage expansion. So all this together make us to hypothesize that maybe here we observe a phenomenon of memory reactivation, and this is why we observe not like a whole phylogeny, but just a part of this, because not all the lineage couldn't interact with the antigen, and maybe such just a part of the lineage was activated. So to check this, we searched for some examples that maybe there are some lineages with the intermediate features between HBMM and albumin clusters. And we found one example with a clear transition of the state of HBMM cluster to the albumin. Here we observe that all clonotypes of antibody-secreting subsets, they form a monophyletic clade on a tree, and all they came from a single time point. And if we would consider the root of this clade, it would be the most recent common ancestor of this clade. It would be distant from the germline. So if you would just sample this part of a tree, we would classify it as an albumin lineage. However, another part is absolutely has features of HBMM cluster. Here, memory clonotype sits on longer branches and came from 2 time points. And the MRCA of this part of the lineage is almost matched with the germline sequence. So in at least part of cases, we observe we can say that albumin lineages, they can arise from the reactivation of HBMM lineages. So next we created the explanation why we observe most albumin lineages just in a single time point, because HBMM lineages represent persistent memory, and the memory can persist in various repertoire frequencies. So we sample just the most abundant lineages, and most of them persist on frequencies which is lower our detection limit. So this is why we observe them only in the time point of clonal expansion. And here in the case presented on previous slide of HBMM to albumin transition, we sampled the lineage persisted on close to the detection limit, so we sampled it in different states. So next we wondered what's the evolutionary mode stand behind the development of these 2 types of lineages. And first we focused on this part of evolution which happens between the germline sequence and the divergence of observed clonotypes in the lineage. So to do this, we adopted McDonald-Craitman test, where we considered somatic hypermutations accumulated between the germline sequence and the most recent common ancestor as a divergence substitutions, and normalize them on polymorphisms accumulated after the divergence of observed clonotypes. So the joint diversity of both HBMM and albumin clusters revealed the signature of positive selection on this part of their phylogenies. However, the signature was more pronounced in albumin lineages, and it was clear for joint diversity and both for comparison of individual lineages. So next, we went deeper to the part of evolution which occurred after the divergence of observed clonotypes, and compared non-synonymous to synonymous ratio of polymorphisms between 2 clusters. And here we revealed that both of them have, both clusters has a signature of negative selection, but in a HBMM cluster, it is more relaxed. So then we look deeper on this further evolution after the MRCA, and also used for this site frequency spectrum, which represents the distribution of somatic hypermutation frequencies accumulated in the B cell population. So comparison of site frequency spectra revealed that albumin lineages has an excess of high frequent somatic hypermutations. However, these effects can be explained by different processes. Demography also can affect high-frequency spectrum, and as we observed on lineage dynamics, albumin lineages underwent clonal expansion during the period of our observation. So it can be also explained by demography. So to distinguish selection processes from demography, we compared again non-synonymous to synonymous ratio of polymorphisms in different frequency bands of somatic hypermutations. So if demography equally affects non-synonymous and synonymous variants, selection acts only on non-synonymous ones. So, and here we again observed the excess of non-synonymous somatic hypermutations in albumin lineages, and concluded that they are reached, say, high frequencies forced by positive selection. So summing up all this together, we came for the following model of what we actually observe. HBMM lineages represent some persistent memory which is in a waiting state and do not interact with the antigen. It do not actively produce antibodies because it composed from memory phenotype with the IgM isotype. However, on the distance between the germline sequence and MRCA, we observe the sign of positive selection, which is more likely is associated with the primary activation of the lineage and the primary affinity maturation in which the lineage was involved. And in the further evolution after the MRCA, we observe here relaxed negative selection, which most likely allows the lineage to restore the structure of B-cell receptor in the waiting stage for the next antigen encounters. In albumin lineages, we observe different picture. It's an actively producing antibodies lineages because they are composed of plasmablast and plasma cells and have another switched isotype. And here, in addition to the sign of primary activation and primary affinity maturation, we have a stronger signature of positive selection, which is most probably is associated with the involvement in neurons of affinity maturation during the reactivation. And here, after the MRCA, we see here strengthening of negative selection and again the sign of positive selection, which again indicates the involvements in affinity maturation, because it occurs in both directions. So beneficial variants are favored by positive selection and deleterious ones simultaneously are eliminated from a negative selection. So for the best of our knowledge, this is the first time when phylogenetic analysis revealed and proved that affinity maturation can occur again after the lineage reactivation. So this was the first chapter, which considered the host pathogen universe from the side of host and adaptive immunity. And the second chapter is devoted to the intrahost evolution of the pathogen on the example of SARS-CoV-2 virus. So as most RNA viruses, SARS-CoV-2 rapidly evolves, and its evolution is characterized by consequent emergence of lineages or variants of concerns, which soon after their emergence outcompeted all other viral diversity present in that moment. So such evolutionary dynamics indicates that virus adapted rapidly to the human population. So many ways of such adaptations were well studied. For example, some variants which increased viral transmissivity and escape from neutralizing antibodies. And the interesting thing that many mutations occurred in a viral population, they are highly recurrent. So they independently emerged in several SARS-CoV-2 lineages, as well as in individual patients with a prolonged COVID infection. So in this context, immunocompromised patient became of particular interest because usually they have a much longer period of viral persistence, and evolutionary rate of viral population inside them is higher, and you can track these changes and perform experiments to understand their effects in the host. So during the pandemic, a whole bunch of case reports were published where case reports of immunocompromised patients with a prolonged infection, and in some of them, authors reported and validated their immune escape from broadly neutralizing antibodies. So this chapter is also devoted for the case study, our case study of patient S. And this case study stands out among other because of several reasons. First of all, it was the longest case of viral persistent known on that moment, and the number of detected changes which viral population accumulated in patient S was pretty high as well. And another important difference is that our patient was almost absent of B-cell immunity, because in addition to rituximab therapy, which targets B cells, it also got the convalescent plasma treatment in the very end of our observation period, when in other patients, usually all these patients received this treatment and had some humoral immune response from other recurrent patients. So patient S is a female with a non-Hodgkin's diffuse B-cell lymphoma of stage 4, and symptoms of COVID infection were first detected in her in April 2020 and lasted almost a year. And she had 2 periods of pneumonia and several periods of PCR positive and negative tests. And these first symptoms appeared after the contact with patient A, with which patient S share a hospital ward. So we collected samples from patient A and from patient S. Here were swab samples for viral sequences and blood collected for flow cytometry. And in the very end of this observation period, the patient got the treatment by convalescent plasma and underwent autologous hematopoietic stem cell transplantation. So during the whole period, the patient received the rituximab therapy in different combination with other drugs. So in this work, we sequenced viral genomes and performed phylogenetic analysis and analyze the effect of these mutations on antigen presentation of viral epitopes, perform some experiments, and extrapolated them on a population level. So I say we here, but actually my contribution in this work is computational and is focused here on the analysis of mutation effects on antigen presentation and extrapolation them on a population level. So first, let's look what phylogenetic analysis revealed. The samples from patient S formed monophyletic clade, which is placed here on a B11 general phylogeny of SARS-CoV-2 virus. And the sample from patient A, the probable source of infection, was en route of this clade. So here we proved that it was a single infection and not a consequence of several infections, and proved that the patient A was a source of this infection. We detected altogether in all these samples about 40 changes in a viral genome listed here, and among them there were several famous ones, such as delta F combination, which is famous because it is associated with an outbreak in mink farms. However, here is the mink associated cluster, and these delta F combinations appeared here in this in our patient independently from it. So evolutionary rate in our patients was almost twice higher than in the general population. So changes which we detected had different dynamics. Some of them were fixed in the earlier time points, some of them were fixed later, and some of them were replaced by other variant or by ancestral variant. The interesting thing was here that all these mutations were more or less equally distributed between different viral proteins, when usually in another analogous case reports authors showed that they are mostly accumulated in the spike protein, and explained that by immune escape from neutralizing antibodies. So such a distribution of mutations made us hypothesize that maybe a T-cell immune response is present here, because T-cells targets any antigens independent of their position in a viral particle, and B-cells targets them only on the surface of the virus. So to check this, the following pipeline was developed. So we considered all positions which were changed by mutations in the virus, and considers all possible peptides of corresponding length for binding to HLA alleles, which cover this changed position. Next, we predicted the rank for this peptide of its binding affinity relative to the distribution of natural random peptides, and have chosen a best rank among all these peptides. And this is how we got the BR score, which represents the ability of some position to be presented on some particular HLA allele. In cases of some positions, there were also known immunogenic epitopes which evoked T-cell immune response in COVID-recovered patients. And in such cases, when the position was covered by such immunogenic epitopes, we also calculated immune BR score, where best rank was chosen among these immune epitopes. And also to estimate the overall ability of some position to be presented on all alleles, HLA alleles of the patient of some class, we calculated the harmonic mean of these BR scores, and this is what the PhD score. So let's look what we observed. So comparison of PhD score of mutated position in ancestral state and derived state revealed that in HLA class 2 alleles there were no significant notable shift in binding affinity of viral epitopes, changed viral epitopes to HLA alleles. However, in HLA class 1 allele, we observed that some position really decreased the binding affinity of epitopes. So we also compared the position which were covered by some known immunogenic epitopes. There were 12 such positions, and they were found only for HLA class 1 alleles, and compared their immune BR scores. So here we observed that binding affinity significantly decreased after mutations in such immunogenic epitopes. So to validate these predictions, our colleagues from St. Petersburg performed some experiments. So first we focused on the position which had the highest fold change in the PhD score, this one. And it is also interesting because this position changed twice, actually. The first variant was not fixed, but reached high frequency, and the second one replaced it in time and was fixed in a viral population to the end of our study. So T-cells, cytotoxic T-cells from the patient S was stimulated by 3 variants of these peptides, by ancestral variant and with the first substitution and with the second substitution. And we revealed that the ancestral peptide evoke a notable response in patient T-cells, and the first substitution evoke a little bit weaker response. However, the difference is not significant here, and the peptide with the second substitution evoke no response at all, so clearly indicating the escape effect of this variant. It was in an earlier time point, in the first one, and the second time point after autologous hematopoietic stem cell transplantation, there was no response for any of these variants, most likely due to that transplantation restructured the whole structure of T cell repertoires. So next we performed analogous experiments with the mixture of peptides changed by these 5 mutations. All of these variants were fixed in a viral population to the end of the study period, and we observed the similar picture. So the mixture of ancestral peptides evoked a strong T cell response in of T cells, and the response was absent when peptides were changed by the mutations. So again, shows a clear escape effect. So summing up these 2 experiments, we estimated that acquired mutations, which we checked experimentally, they resulted in a loss of as much as 1% of effector CD8 T cell population, which is this number is actually comparable with the overall number of responding T cells usually in a patient during viral infections. So next, we wonder how this escape effect of patient's variant can be applied to the general population. So for this purpose, we considered all the set of most frequent HLA alleles globally, which altogether cover the 95% of diversity in the human population. And here alleles of patient SI are marked by a red color. So we revealed that this escape effect for patient S allele would be quite surprising if you would randomly choose some set of HLA alleles from the general population. So this escape is quite specific for the patient in HLA class 1 alleles. However, these alleles are actually very frequent globally and in different populations, and in some mutations there are other alleles with comparable effects of change of binding affinity of this mutation. The picture was different with the HLA class 2 alleles. Here, the effect of mutations were in general much less pronounced on the antigen presentation, and there was nothing surprising in patient S alleles in comparison with other alleles in the general population. So we also compared the overall effect of all mutated positions on different HLA alleles, and here we again revealed that it really depends on a particular HLA allele and can be very different. So, of course, this study has certain limitations. Unfortunately, we were very limited in the amount of blood collected from the sample in an earlier time point where we really could test it, the escape effect of viral changes, and we cannot, in the experiment with a mixture of peptides, we cannot distinguish which is it all of mutations which have escape effect or just some of them has, and maybe some of those peptides didn't evoke immune response even in ancestral state. Also, we didn't tested experimentally the effects of other mutations which were not used in the experiment, and we did not study here the way of immune escape when the virus do not change the binding ability or the binding of the epitope to the HLA allele, but change the binding of epitope to the T cell receptor. And here we relied on binding prediction through HLA alleles. However, Anyway, we revealed a clear immune escape from cytotoxic T cells, and it was the first time where it was shown the immune escape from cytotoxic T cells can be a driving force of SARS-CoV-2 evolution. And actually, this work arised many questions how this force is acting in the general population, and now we continue to investigate this question. So here are my general conclusions about these two chapters, and the most general conclusion actually is that intrahost evolution of both pathogens and adaptive immunity should be always considered with keeping an eye on the other side of this host pathogen universe, because they are really dependent. So thank you very much. This is publications where I participated, devoted to these topics, and I want to thank the group of Professor Bazikin, and I will say thanks in more details later. So I think I'm finished.
Okay, and one thing that I forgot to do as usual was to present the candidate herself. So by now, everybody has got the idea what was she working about.
Who is it?
Who is her? Who is she? Yes. So I'll just mention formal things. Evgeniia graduated with honors from the Department of Human and Animal Physiology at the Biological Faculty of Moscow State University, and then joined Skoltech and got master's degree in bioinformatics here, again with honors, and joined the same year she joined the research group of Professor Bazikin as a PhD student. So what we've heard now is the result of this study. She has published 4 papers that form the base of the thesis. In 2 of them, she is one of co-first authors, and the other 2, about SARS-CoV-2 evolution, she's a co-author. And her research was supported by grant, so-called PhD student grant from the Russian Foundation for Basic Research. Okay, so now we are done with that, and we shall start with questions from the jury. And again, I think we'll start with the online members. So, Dr. Bagayev, please. Alexander?
Hello. Yeah. So one of the question that I have I got now while listening to this particular work is about initial clustering that you perform to determine two subgroups. Can you tell a little bit more about how you got this group, on what data you basically combined? Which data points to initially that's to separate between these two subgroups that you analyzed in the first part of the project?
You mean clusters of B-cell clonal lineages, right?
Yes.
Can I go back to the presentation, please? So actually, to reveal these lineages, we joined together repertoires from 3 time points, which we had, and repertoires from 3 cell subsets. So in the lineage, we have simultaneously clonotypes which came from different time points and which have different B-cell phenotype. So because of that, we were able to calculate the fraction. Let me find this slide. Okay, here's it. So because of that, we were able to calculate the fraction of memory B cells, plasmablast, and plasma cells in the lineage, and in the same time fractions of IgM, IgG, and IgA isotype, which we were able to distinguish in a repertoire. So actually, this is just a PCA of lineages based on these three variables, and we performed Kamin clustering to distinguish between these two groups. And the first idea to do this came from just comparison of distributions of this fraction of memory clonotypes. I have a picture on an additional slide. So here, so just if we compare the distribution of B memory subset fraction in the lineage, we observe here bimodality, which and in the fraction of IgM as a type. So this is why we clustered them and considered them separately.
I see. Did you use specific markers to determine the memory B cell?
Yeah, sure. But unfortunately, I do not remember them exactly because it's not my part of work yet. But it's usual markers which used to distinguish memory B cells and plasmablast and plasma cells, which are were used by our colleagues during the cell sorting.
I see. Yeah, my markers could be really tricky, but the difference is very huge. So we definitely can assume that that's correct. So from my view, I have the minor minor comment is that the previous in the manuscript that I've been reading, the data between two chapters was my proposal was to connect those chapters more tightly. Of course, it's impossible to get back and to do the additional work, but it looks to me very logical to include the B cell repertoire analysis to the second chapter as well, to see the evolution of the B cells during the COVID. But that's what just a comment.
Yeah, it's actually what also Professor Kharmeeva advised in her review. So I restructured the whole introduction part and conclusion to make it more confluent. So I hope it's became better after this restructuring.
Yes, thank you. So the other my minor comments was resolved by Evgeniia. Have several grammatical comments that was fixed after the initial review. Is it possible for me to think more a little bit? I have some notes and then maybe get back to the additional questions after the.
Well, everything is possible. Okay, go ahead.
Thank you. No, I mean, I need to think a little bit about additional questions, and then is it possible to ask questions after the other members of the jury?
Definitely, yes. Just proceed.
Or shall we move just to the next jury member, maybe?
Yeah, probably. It looks like something is wrong with the connection. Okay, so could you return to from Elena? Could you please return to the view of jury members? Okay, so yeah. Okay, then Dr. Yefimov, please.
Okay, yeah. First of all, my congratulations to Evgeniia. It was a beautiful work and excellent presentation. Then I have a couple of questions, all of them directed to the second part, to the T-cell response to the SARS-CoV-2 and evasion of the virus. So the first one is, correct me if I'm wrong, you've identified 31 mutations which affected amino acids, which were with amino acid changes, and looked to the immunoepitope database for the known T-cell epitopes presented in the patient's HLA alleles, and you found 17 epitopes which fall onto the 11 of these 31 mutations. Is that right?
Yeah, I think it's right. But when we considered immunogenic epitopes, we filtered them that the immunogenicity was shown on some H allele which has the patient as has. So these mutations altered some other immunogenic epitopes which like were experimentally validated on other HLA alleles or has no specific specificity of the allele or marked in the database. So we didn't include it in other analysis. So
Okay,
we used just specific HLA epitopes.
Yeah, so here comes the question then. Did you check how many were in general in the IADB database, the epitopes which would be presented with using this exactly the same criteria, which would be presented by the patient HLA alleles? So the question is, how much more than those 17 there would be?
It was
theoretically recognized.
Yeah, I got the question. There were some more, but they was not changed by mutations in the her virus during the study period. So actually, this work was like done about 2 years ago. So maybe there are more epitopes published today, and it can be more in a database. But as far as I remember, to that moment, maybe it was twice more than we used in other study, maybe like 35 or something.
Okay, yeah. And of course, it doesn't mean that they were actually recognized in this patient. Okay, so then the second question, I'm not really sure about your interpretation of the immune response in the second time point. You've showed for your major epitope, mutated epitope, and also for the 5 additional epitopes that they were not only mutated epitopes were not recognized at the second time point, but also the wild type epitopes were not recognized. But then you also tested this panel of peptides provided by Generium, and I assume you don't know the exact sequences. And you, in the text, you said that you were able to see the response in second time point, but the figure itself for CD8 T cells, I think it's not really convincing that there was a response. What's your actual interpretation?
Sorry,
was
maybe
there?
Maybe I said something wrong in my speech. We observed no response in the second time point at all. So, like, it can be maybe because these T cell clones were not needed anymore. They really decreased in their frequency in repertoire, and we didn't detect it the response. And also the transplantation should really restructure T cell repertoire. So that's like it's not quite surprising.
So even for this panel of other epitopes, you didn't see any CD8 response.
Yeah. So we performed absolutely analogous experiments. Not we, like our colleagues performed absolutely similar experiments for both time points, for early one and the later one, and there were no response in the later time point. However, the cytotoxic T cell population were normal there, so in the blood.
Okay. Thanks. And my last question is a bit more general or philosophical. So you saw this at least six mutations which might have been recognized, but you can assume that at least one of the 5 were recognized, right, at the first time point, and then it was mutated, and the mutated epitope was not recognized by the T cells from the same time point. But also from your mutational analysis, it seems that the mutations didn't occur simultaneously, and it would be a strange coincidence if they did. So the question is to you, do you think that even partial evasion of the immune response is somehow beneficial to the virus? So if it presents, I mean, definitely more than one epitope is presented on the surface of the infected cell, but if you decrease the number of epitopes presented on the infected cells, would it, in your opinion, give the virus some kind of survival benefit, so it can evolve even further and mutate more and more epitopes?
It's an interesting question. I think that if infected cells would present less number of epitopes, the selection force will stronger on this position, become stronger. So maybe it would be less time required by virus to accumulate changes which make an escape of these epitopes. When like the number of epitopes is larger, so it's have more ways to escape immune response. Or I got your question wrong.
No, I think you got it right. So yes, okay. The question is, if one infected cell presents 5 different epitopes and the other infected cell presents only 4 different epitopes, would the second one get some advantage? Yeah.
Actually, it's really interesting what's the dynamics of this an escape. We cannot take it from our data, so we do not know whether it was the consequence of events. Maybe like it was one epitope was ruined, and then adaptive immune response developed new T cell clone for a new epitope, and maybe it was not happening simultaneously. Maybe it was a consequence of escapes and new T clones involvement.
Yes, you're right. In principle, it could be the case, but what we saw in our data that in the majority, or even in all of the patients, more than one epitope is recognized at the same time. So it would be, but of course, it's different patient with.
You mean usually not a lot of epitopes involved in the immune response?
Usually more than one epitope is involved.
More than one, yeah.
Yes,
yeah.
Exactly.
It should be logical.
Okay.
Yeah, to use more epitopes. Thank you.
Okay, thanks for your answers. And yeah, my comments have been addressed in the changed version of the manuscript.
Okay, and I forgot to ask the same from Dr. Bagayev. So, Alexander, are you satisfied with the responses and changes in the manuscript for the record?
Yes, I mentioned that my minor comments was.
Okay. Okay, now Professor Neher.
Yeah, thank you very much for your very interesting talk. I have a number of questions on both chapters, and some of them might be a little speculative, so it's totally fine if there isn't a definitive answer to them. But I'd be all the more interested in hearing if there is one. And my first question was actually also already answered by Dr. Bagayev on the clustering. And if I understood this correctly, so you're clustering on cell distribution and type, IG type.
Mm-hmm. Yeah.
So the first observation then that these lineages that you identify in HBM and LBM, that they differ in these quantities, is then not so surprising, right? That's something that you basically put in, right? But the sequence features, they didn't go into the clustering, right? So this is kind of an independent event, if I understood, independent observation, if I understood correctly.
Yeah.
Yeah, thank you. So then the first question I had, so your time points T1 to T3 are quite unevenly spaced, right? There is sort of a 1 month between the first two and then 11 months between the latter. Is that reflected then also in the patterns of persistence? Would you expect more things to persist from T1 to T2 than from T2 to T3 because the gap's much larger?
Actually, such sampling came from the history of all this study, and so I didn't have time to tell all these details. Initially, our colleagues, they wanted to study allergenic response on the pollen, so they sampled this blood before the pollen season, and after the pollen season, and like in a year before the next pollen season. So that's why we have these uneven periods between time points. However, when we started to analyze the data, we revealed absolutely no difference. There were also like allergic donors and healthy donors, and there were no difference between them in the peripheral blood, or it's maybe it's not enough size of immune repertoires to detect this difference, or not well designed the experiment to detect such information. So we just joined them, and since we observed no difference, analyzed them as a healthy individuals. So among that 4 healthy individuals, 2 of them are really healthy and 2 of them have this pollen allergy. So this distribution didn't come from any expectations of lineage persistence or something.
Yeah, no, I understand that. My question was whether there is any, if you look at persistence, you'd expect maybe to see more persistence over a month than over 11 months, right? Is there sort of any hint of that? That maybe an expanded LBM clone might persist over 4 weeks, but not over.
Maybe it would be different to make higher resolution in these dynamics. But I really think that HBMM lineages, they stably persist. Whenever how many times you will sample that because these persisting large lineages were described in several other studies with various designs of repertoire sequencing and various dynamics of sampling. So, but with the albumin lineages, it's really interesting, how they continue to develop in time.
So my next question was on the McDonald-Kreitman type tests, right? So for one of these 2 categories, you tend to see much longer distance to the germline ancestor. So meaning you have a lot more mutations to interrogate positive selection on, right? To what extent do you differences that you see in the degree of positive selections are due to the power that you have more power to see them in one case than the other, or whether they are intrinsic an actual signal? To what extent is it a power difference or an actual difference?
Yeah. Actually, we also thought about that groups are not even, and the distance, the number of mutation is not even. So I have a table in some supplementary data, and actually have an additional slide. Maybe we can switch. So I performed this McDonald-Kreitman test several times with a different inclusion criteria. So, and the most reliant result, I think, when I considered joint diversity inside the cluster, in HBMM cluster and in LBMM cluster together, all mutations. And of course, in HBMM cluster, there are a much smaller number of mutations, but this group is much larger. So it's in 3 times larger than an LBMM group. So summing up, we observed more or less a comparable number of mutations in the HBMM and LBMM clusters. So yeah, and I also compared it like joining these divergence and independently on different lineages, both excluding lineages which have no mutations in G-MRCA period, or with adding pseudo counts. So like I had several designs of these tests, and results were more or less comparable.
Yeah, thank you. Maybe we'll move on to the second part. So on the tree that you showed, right, these are sort of consensus sequences from, I guess, tiled amplicon, RNA sequencing, right? So RT-PCR amplicons, consensus sequences. But one of the graphs that you showed, you didn't talk much about it, also indicated, positions at intermediate frequencies, sort of variable positions within the host. How much diversity was there? Have you has anybody tried to maybe outgrow different viruses from these samples and investigate the within host diversity a little bit more systematically?
We've thought a lot about this and about this project, and actually, in my mind, there is no like exact publication about such thing. But usually, yeah, if you like take just sequence libraries from the GISAID, there are some diversity in almost any samples. But I just know from I have other publications when it's not a long-term COVID, but a usual COVID, usually no more than 1 or 2 changes is happening in a single patient. So which reach the like consensus frequencies.
Yeah, but that isn't an acute infection, right? You had an infection that lasted a long time, and sometimes there is a lot of diversity that accumulates. And there was a diversity in your case, as far as I understand, from the allele frequency graphs that you've shown.
Yeah.
But all these January 2021 samples, I think they were sort of all very tightly clustered, so they didn't seem to suggest that at that point there was a lot of diversity.
Maybe I didn't got you. So you mean was the diversity inside a single sample?
Well, I'm mostly interested in the diversity within the host, and to some extent that will be a single sample, but might also be represented across multiple samples from similar time points.
Yeah, maybe, but we had replicates, so we like sequence each time points by 2 or 3 times and joined libraries together. So and did not rely on positions which were poorly covered. So.
Yeah, no, that makes sense. Okay, maybe one final question regarding that that HLA escape where you had two subsequent mutations, first one and then the other. Do you know why? Is the first amino acid substitution more accessible by mutation, or has one of them made of a fitness effect on the virus? Is there sort of an explanation why you first find the other one and that then is then supplanted by the second one, other than just the different degree of escape?
Yeah, actually, I searched the literature for all these mutations. That mutation is in a SP3 protein, so there were no information about other effects of this mutation on viral fitness. And I think it's just like it decreases the T cell recognition a little bit, so or decreases the binding affinity. So it has some effects. So maybe we just do not see that much on our experiment, and it can be seen in other experiment designs. So it was seems it was some kind of useful for virus, because it was about 80% of frequency in a viral population, and it was a little bit surprising that such mutation was fully replaced by other variant in the end, but in the end it was like 100 frequency of the second substitution. It also can be a clonal interference, so maybe it's not the deal in this particular mutation, but the another clone itself, which was in general more beneficial.
But both mutations were accessible by a single nucleotide change.
Yeah, there were several deletions, like 3 deletions there, yeah, but most, and 1 stop codon, but most of them were, yeah, single nucleotide substitutions.
Yeah, thank you very much. I didn't have any corrections, so I'm all good.
Thank you.
Okay, and again, the formal question: are you satisfied with changes in the manuscript and with responses to your questions?
Yes, I am, and I didn't request any changes to the manuscript.
We need it for the record anyhow. Okay, thank you. And now we move on to members of the jury here. Professor Ivankov.
Okay, I actually don't have any specific comments or questions, and from my side, I can only say that the work is a very elegant work. It combines the view of evolution from both sides, from hosts and pathogens, and the second case is just amazing. Evolution of pathogen in hosts during 300 plus days in one and the same patient. It is just luck. However, Evgeniia with her colleagues managed to take advantage of this luck and analyzed everything. And so I don't have any comments, as I said before, so I'm automatically satisfied with the absence of the answers. So we can proceed, I think.
Thank you very much.
Okay. Okay, Professor Khrameyeva.
I also think that this work is excellent. However, I had several comments and suggestions regarding the text of the manuscript, but they were all addressed in the current version. So I'm now completely satisfied with the writing of the manuscript. And also I had a couple of comments on the results part, but they were quite minor. I just asked to calculate the statistical significance in a couple of cases where it was missing. Yeah, but this also was corrected. So I'm happy with that too. And regarding today's talk, I have one more additional question in addition to what I wrote in my report. If you could go to the presentation and the slide number 51, please. Yeah, here I just wonder, I'm looking at this figure in the right part of the slide, and in the first 2 columns of this figure, the dates these samples were taken are quite close to each other. It's August 17 and August 20, but the difference between these 2 samples seems to be quite dramatic compared to what we see for the last 4 samples, right, which are quite similar. I wonder how this could be explained. Is there some dramatic effect in the evolutionary sense that happened between these 2 time points, or it's just a quality issue with a sample, with one or another sample?
You're talking about the difference in mutation frequencies or in the coverage between samples?
Both, I think. I mean, if you look at the second picture, at the second part of the picture, the samples, they look quite similar to each other, right?
Yeah, it's true.
both in coverage and
yeah, I have several maybe explanation for this. The first one, biological, so that's maybe it's an earlier time point, and then maybe here like events were more dynamic. I don't know, maybe there are really more fast changes here, and also these two samples and technical reason that these two samples are differently sequenced and differently covered in some regions, and so because of this low coverage, the frequency become more noisy, maybe. So this is why on eye we observe such difference.
So just speaking about the quality of the sample, do you observe any issues with the quality in this first or second time points, or they are more or less the same? Like
actually, unfortunately, I didn't work with this data. I joined the project later, so I didn't analyze the like raw sequences by itself and the quality. But I know that there were several replicates here, so sometimes this like position was not covered by one replicate, but was covered by other. So and they are like presented here by a really small dot, because of this low coverage. And also I need to add that this picture actually shows any detected changes with even a really low coverage, but not all of them were included in the analysis. We had some threshold and didn't test it, the effect of mutations on the antigen presentation, which have this low coverage, and didn't include it in them in like DNDS calculation, which you described.
so maybe it would be interesting and I don't know, needed, necessary to compare replicates for each time point, and for each time point, calculate how similar they are exactly in these time points. Maybe that would help to answer my question whether this is a technical issue or something biological. And if you believe that this is a biological thing, is there a way to calculate an evolutionary rate specifically between these two time points and maybe specifically between later time points and compare?
Actually, there are no many positions where differ between time points. So mostly they like the variant is everywhere detected in all close time points. It's just they're different in coverage, or they're different when here the position was not covered at all. So I can. It's also not very all colors not very well reflected. But as far as I remember, in a close time points, it's just like maybe a couple of positions which really was different. So there was like no need to calculate rate of changes between close time points. Like maybe one or two we can find here. So this one, as far as I remember, is light yellow. This one here, I know.
Yeah, okay, I see. Thank you very much.
Okay, thank
I'm
you.
happy with this presentation.
And as for myself, actually, I've made comments, some comments at the preliminary stage. So by now, I have nothing to add. And indeed, they have been taken into account. I'm also quite happy with that. So if there are no questions from the jury, maybe any second thoughts? I see none. Okay, then there's time for questions from the audience. I see no audience in Zoom, but there are people here, so any questions? Again, see none. Then Professor Bazykin.
So Zhenya's work, I think, demonstrates how useful it is to apply ideas from population genetics to novel fields if that needs any further proof. And for example, her work is, to best of my knowledge, the first application of the McDonald-Kreitman test, which is probably the most famous test for positive selection to immunology data. But beyond that, I think it addresses, beyond methodology, I think it addresses pretty fundamental questions. And not all that she has done has made it into the thesis. There is an unpublished follow-up of the research on this T-cell immunity escape, in which she and others are showing that this T-cell immunity escape is actually playing a role in the global evolution of SARS-CoV-2 worldwide. And Zhenya came from a very different field. She's a zoologist / animal physiologist by training, but she has made gigantic progress over these years, obviously, and she's one of those people that make everything that they do seem pretty easy. And it was, a huge scientific joy working with her, and she has become obviously an independent and mature researcher in the field. And I congratulate Zhenya on her great work and wish her luck with her future endeavors.
Okay. Well, that's it. So it's probably time for your final remarks, if you have any.
Acknowledgements or the
acknowledgements, disacknowledgements,
okay.
acknowledgements of evil done to you, whatever.
I just forgot when it should be said. Okay, thank you very much. Yeah, I have something to say, of course. So first of all, I want to thank all jury members for their participation in this defense procedure and their valuable feedback on my work. And I also want to thank Nadezhda Dansu and Yelena Dito for the organization of this event and the valuable help in the preparation. And this work would be impossible actually without the help and support of a lot of people. So first of all, I'm deeply grateful for my supervisor, Yegor Bazykin, who gave me the opportunities to spend my PhD years in his group. And I'm very thankful for Yegor for sharing his expertise and experience, giving freedom in my scientific search and in a way of thinking, and friendly work atmosphere and human support in various situations. So behind a cool chief, there is a cool lab. I also want to thank all Yegor's group, which is a very friendly and warm team, where you always feel a pleasure to be. So it is not just a group of nice people, but a team with a high level of scientific integrity, where you always can get honest feedback on your work and find inspiration to try something new and ask new questions. So I started to list lab members, which I want to thank the most, but actually I realized that I'm just repeating the list of the lab from the website. So I will not do it. And I thank you all, guys. It was a pleasure to be with you these years. And I really hope that all of us will be safe and in touch, and a lot of interesting projects and collaborations are waiting for us to be done together, in spite of growing distances and circumstances turning out our lives together this year. So I'm grateful to my co-authors for our work together, our shared ideas and experience. For the project of B-cell clonal evolution, I especially want to thank Artem Mikhailov and Ivan Zvagin, who designed the study, got the data, and were really open-minded about various ideas, how to do different evolutionary stuff on their data. So I'm happy now that these computational experiments finally resulted in a beautiful story about the work of B-cell immunity. For the case study of long-term COVID-19, I would like to express my special gratitude for Anna Polina Shurigina and Maria Sergeeva, who validated our computational conclusions through laboratory experiments. And I'm also very grateful for Mikhail Shugay, Alexey Neverov, and Dmitry Chudakov, who gave their valuable feedback and advices during the analysis in both projects. All this would be impossible without Alexander Tashkeev, who fascinated me to study bioinformatics together and encouraged me to enter the MS program in Skoltech. I'm very grateful for him for the support, inspiration, and a lot of help on this way. I also want to thank Yaroslav Ispalatov, my first supervisor at Skoltech, who guided me in my first computational steps and in general greatly expanded my worldview. And I'm very grateful for Skoltech in general, for its community and the high level of science they're doing. And of course, I'm very grateful for my beloved family and friends for the huge amount of love and support they gave me in this way. So.
Okay, thank you. And now I probably have to ask the audience to leave because the jury would discuss the outcome. Okay. Alekseeva. Okay, yeah. Dr. Alekseeva, my congratulations. You've done a great job. And now it's tears time.